morning. This is Tim Guilford, your glutathione guru. And today we're going to talk about the role of glutathione in COVID-19, both the acute form and the chronic form. But I'm available for discussion of any of the information here. And much of what I'm going to say can be found at readysorb.com/COVID. There's my email contact, and you're welcome to contact us by telephone. And I'm happy to answer any questions or observations you may have. When I talk about glutathione, I usually start with a picture of the molecule of glutathione and its three component amino acids, glycine, cysteine, and glutamine. They've been formed in a unique form that resists breakdown by most enzymes in the body. The active form in glutathione is this sulfur molecule, which can donate a hydrogen and its electrons. To facilitate the function of plain glutathione, we encapsulated glutathione in a liposome, and this allows a molecule that is readily absorbed, and that's why we named it ReadySorb, by the way, and it increases its function. As I mentioned, glutathione can be broken down by one enzyme known as gamma glutamyl transferase, or GGT. GGT breaks the molecule down into its component of amino acids, which can then be incorporated inside the cell and reconstituted by the means of two enzymes back into the form of glutathione. The advantage of the liposome is that the liposome containing glutathione can be absorbed directly into cells, and then the lipid membrane stripped off, allowing plain glutathione to become available for the functions of inside the cell. Glutathione is transported by two independent pumps into the mitochondria, and this means that the free radicals that are formed during energy production can be neutralized by glutathione that's been absorbed from the cytosol, from the surrounding area inside the cell. This is done, glutathione is constructed with the means of glutamine cysteine ligase and glutathione synthase, and maintained inside the cell. As far as free radical function is concerned, glutathione can donate a hydrogen and its electron directly to the hydroxyl radical, for example, and then neutralize it to harmless water. Enzymes also work with glutathione, and there's a whole symphony of enzymes that work with it, such as the peroxidase enzyme, which brings glutathione in closer contact to the hydroxyl radical and increases the speed of the neutralization to harmless water. This happens in our cell over and over, thousands of times every day. The production of glutathione can be stimulated when there's an excess of oxidation stress in the interior of the cell. This releases a material called NRF2, which is the oxidant stat inside the cell, which says your body needs more antioxidant. It travels to the nucleus, where it stimulates the formation of what's called the antioxidant response elements, which includes glutathione, as well as the symphony of enzymes that I mentioned. It's little known, but it turns out that NRF2 needs a component called BRCA1 to have the most efficient function. When BRCA1 is not available, there's a decreased production of the antioxidant response elements and glutathione. Now, many people have heard of BRCA1 in other scenarios, and we'll talk about that in a moment. Let's talk about the S protein, the spike protein of COVID-19. We're all familiar with this diagram by now, which shows the spikes of COVID-19. And these spikes will bind to the receptor on an element called ACE2, which facilitates the absorption of COVID-19 into cells. ACE2 is found in many parts of the body, including the nose and pharynx, and in the lungs. In the lungs, here's a picture of an inside the lungs and an air sac called an alveolus. And you have both type 1 and type 2 pneumocytes, as well as macrophages patrolling and keeping that area clean. Turns out that the macrophages inside the alveolar lining of the cell pick up glutathione from the extracellular lung fluid that is expressed into the surface of the interior of the air sac. This turns out to be a critical component of the macrophage, which is important in maintaining a clean and non-inflamed environment. In the type 1 and type 2 pneumocytes, type 2 has an ACE2 receptor, which can bind with COVID-19 and cause damage to the cell. When these cells are damaged, they in turn can no longer take care of the type 1 pneumocytes. Type 1 pneumocytes become swollen, and this is the problem with gas exchange, because gas exchange occurs with the type 1 pneumocyte. It turns out that the exchange of gases such as CO2 and oxygen is important, and our shortness of breath is determined by the ability of CO2 to get out. So even when O2 cannot get in as well, people will feel as if there's no problem with air hunger and not feel short of breath. And this is why people could present to the emergency room in apparent reasonable health, feeling bad enough to go to the emergency room, but still able to communicate with speech, for example, and then a few minutes later require intubation because of an extremely low PO2. In these cases, the air hunger drive was not being expressed while O2 was becoming extremely low. It turns out we now have the advantage of knowing that there are 4 clinical studies that document deficiency of glutathione that occurs during COVID-19. This was first documented in May of 2020 in a small article by Polonikoff from Russia. Polonikoff felt that the endogenous deficiency of glutathione was the most likely cause of serious manifestations and death in the COVID-19 patients. Endogenous refers to the production of glutathione inside the cells. And I'll show you studies later where individuals had a diminished endogenous level of glutathione that could be replenished with liposomal glutathione. These were in other viral diseases other than COVID-19. But now in March of 2022, we have the advantage of 4 clinical studies, each one of which became increasingly large and showed additional information about the role of glutathione in this disease. For example, the Kurikoff study, the second one in this list, shows that as the reduced form, that's the active form of glutathione, goes down, a material called D-dimer is elevated. D-dimer is an indication of increased thrombus formation and has formed itself as thrombus material is broken down. That's a critical factor in many of the complications of COVID-19. So it looks like it clearly shows that as glutathione goes down, D-dimer is elevated. And platelet aggregability has been shown to be a prothrombotic feature of a number of diseases, including type 2 diabetes, which I will show you is also a low glutathione condition. The study by Laga and Amaral, number 4, shows clearly that the macrophage type cells called peripheral blood mononuclear cells are deficient in glutathione during acute COVID. And this study shows the control levels on the left, with the red colored squares on the right being the acutely ill individuals, and they have a deficient level of glutathione that has a high level of statistical probability of being a significant difference. It's a very high p-value or indicating significance. It turns out that even in mild to moderate symptoms, individuals were shown to have a deficiency of glutathione for at least 50 days after the acute episode. Again, there's a high level of statistical probability of significance in this study. It was also accompanied by a study that looked at the elevation of IL-6. IL-6 is an immune hormone called a cytokine that has been shown to be a standalone indicator of the severity of disease with COVID-19. And here we show the controls on the left, and on the right, the individuals with COVID-19. Again, they show a highly statistically significant difference between the two groups. The intriguing thing is that IL-6 remained elevated for at least 50 days after the acute infection. IL-6 was featured in a paper that I was able to co-author in September of 2020. That was published in September of 2020. And shows that glutathione is made deficient in a number of situations, starting with the S protein. And then as the infection progresses, the N protein that helps the virus replicate actually programs infected cells to produce immune hormones like IL-6. Another immune hormone called transforming growth factor beta is also increased and looks like a standalone indicator of severity. But IL-6 has gained the most notoriety as an indicator. It also turns out that IL-6 can deplete glutathione in cells directly. And this is where I think a lot of the problem with COVID-19 arises, and may explain why COVID-19 has led to severe illness, especially in individuals whose enzymes are not able to make glutathione efficiently, as occurs in diabetes. It turns out that in diabetes, the severity of COVID-19 is tripled, as has been documented in a study. And it's also intriguing that the onset of diabetes can occur post-COVID, even after mild acute episodes. So it's really fascinating to look at as research begins to look at the role of diabetes in COVID-19. Much of this information has been discussed in two studies. And here, this number refers to the PMID number, which can be used to identify our paper just by plugging that into a search engine with PMID in front of it. And this paper explaining additional information was subsequently published in 2020. And you can find that by plugging in the word encyclopedia and the numbers 6357 into a search engine. And this gives you a review of the information I just mentioned. It turns out that in January of 2022, a paper was published that shows that increased levels of glutathione and its antioxidant function will prevent the binding of the S protein from COVID-19 to the ACE2 receptor. And this may explain why some people seem to be immune to contracting COVID-19. Now, I mentioned earlier that the S protein can inhibit BRCA1. And this has been shown in a paper that shows BRCA1 S protein inhibiting BRCA1 in the nucleus of cells. So that as that occurs, the antioxidant gene expression of the antioxidant elements is diminished. So that will end up decreasing the level of glutathione available to cells, and begins to explain an additional loss of glutathione in cells. It turns out that the antioxidant response elements and glutathione are important in DNA damage repair. And that's why BRCA1 has become associated with some inefficiencies in the production of BRCA1 and due to genetic mutations has been associated with certain cancers. So when glutathione is decreased, a wide range of problems may occur. And this was first pointed out in a paper by a fellow named Ned Ballatori published in 2009. Dr. Ballatori was nice enough to include our early paper on liposomal glutathione and the occurrence of atherosclerosis and diminishing atherosclerosis in a mouse model of atherosclerosis. But subsequently, other studies have shown that deficiency of glutathione will predict cognitive decline in people as they get older, even in apparently healthy individuals. And prior to that, a study was shown that a biomarker that consisted of the use of the level of reduced glutathione, as glutathione goes down, the increased risk of mortality occurs in people with coronary artery disease. And this begins to explain the difficulties people see with brain function and cardiac problems post-COVID-19 as their glutathione has been depleted. We now have 4 clinical studies showing that liposomal glutathione can restore the level of glutathione in both serum and inside cells. And these studies were conducted in individuals that had chronic low glutathione, such as HIV, which is, as you know, a virus, and also in type 2 diabetes, which is a metabolic disease, which is quite prominent. Children with autism were also studied and showed that their glutathione could be restored. The clinical studies were preceded by cell studies that demonstrated that Bernisorb liposomal glutathione will restore glutathione levels and macrophage immune defense 1,000 times more efficiently than N-acetylcysteine. By this, these studies showed that NAC required an amount of 1,000 times larger than the liposomal glutathione to obtain the same result. So in summary, we have shown, or it's been shown, that as the active form of glutathione goes down, IL-6 will become elevated, and this creates a vicious cycle where glutathione is not formed normally. And replenishing glutathione will actually, in the studies that I just mentioned, restores IL-6 and TGF-beta to normal levels in these individuals with HIV and also in type 2 diabetes. The type 2 diabetes study focused primarily on IL-6. Here the studies are listed, the 4 studies. And again, these can all be identified at bernisorb.com/COVID, so you don't need to worry about writing numbers down here. As a rule of thumb, in terms of dosing, I recommend that if you've been ill for a while, you should start lower with the amount of glutathione, because some people experience what's referred to as a Herxheimer or a detox reaction as they begin to mobilize toxins. If this happens too quickly, people can feel poorly for a little bit, but starting with a lower dose, like a 1/4 teaspoon, and then increasing as tolerated up to 1 full teaspoon once or twice a day has been tolerated very well. We can talk to you about additional approaches if needed. In the acute disease, I don't hesitate to offer 1 or 2 packets 2 or 3 times a day to people with acute COVID, but this has only been anecdotal experience, and I can't quote studies in this regard. So I appreciate your paying attention to the information today. And if you have any questions, don't hesitate to contact us. I'll leave this slide up for a moment so you can write down the contact information.